Hello, I hope everyone is doing well today. We're about to get started. We have a great webinar for you today presented by biotech application scientist Pete Brescia. Pete will be walking us through his presentation on DNA quantification, multi-volume, multi-mode analytical methods. At the end of the presentation, we will be fielding questions which can be submitted with the chat feature in the bottom left-hand corner of your screen. As a reminder, all registrants will receive a link to the recording of today's webinar. Thank you again for joining us, and I will pass things off to Pete.
Great, thank you. Hello and welcome. I want to thank you all for attending the presentation this afternoon. Today, I'd like to discuss methods that will help increase throughput and efficiency when quantifying biomolecules in conjunction with microplate instrumentation. In particular, I'd like to focus on quantification of DNA using a variety of sample and reagent volumes, as well as a range of analytical methods requiring various detection modes. The presentation will also include methods for assessing the purity of the sample with a focus towards identification of some of the more common residual contaminants from sample processing. These can include other biomolecules as well as organic and inorganic compounds that may be present during sample purification and preparation and then used for downstream applications. I'd like to start with a brief introduction to multi-mode analysis and detection methods available for use with a microplate reader for those of you who may not be familiar with microplate technologies. Microplate technologies are most well known for the ability to perform a variety of assays in a high-density microplate format to allow increased sample throughput. This is achieved by the ability for the reader to accommodate an array of vessels with a standard footprint. Commonly used microplate densities range from 6 to 1,536 wells per microplate, with each microwell capable of holding volumes ranging from tens of microliters to tens of milliliters. The 96 well density microplate format is the most commonly used format, and the assays performed are typically biochemical or cell-based in nature and can cover a broad range of analytical methods as well as a variety of detection methods, which include absorbance, chemiluminescence, and fluorescence modes. However, the instruments have been used in a range of industry areas such as food, water, and petrochemical analysis as well. Historically, the most common assay performed is the ELISA assay, which most folks are familiar with. And by using a microplate reader in conjunction with a multi-volume plate, which I'll discuss and is pictured here in the middle of the slide and the lower right, the functionality can be extended to include microvolume analysis as well as accommodation of a 1 centimeter path length vessel, such as a standard cuvette, which is shown in the image as well, or to biocells, which is a vertical path length vessel as well. The ability to perform microvolume assays allows for preservation of precious samples, and this in comparison to the more traditional vessels which require larger volumes. Today we'll focus on absorbance and fluorescence measurements for the quantification of double-stranded DNA, and we'll use several vessel types, including the microvolume analysis. Shown here is a comparison of the various vessel formats showing how the path length is determined for each type. As you can see, both the microvolume and biocell vessels rely on a fixed path length, thereby simplifying the calculations relating concentration to absorbance measurements as seen in the equation. I'll discuss this in more detail in just a moment. However, I'd like to talk about the microplates, which on the other hand have a variable path length and thus require additional methods to determine the path length. Path length will be dependent on the assay being performed or the total volume and the dimensions of the plate wells. Software such as BioTek's Gen 5 data analysis software can determine the path length for you, and I'll discuss this in more detail in just a moment as well. Once Gen 5 has determined the path length, that value can be used to normalize measurements to 1 centimeter path length values for use in accordance with the Beer-Lambert law. I'll discuss both the Beer-Lambert law and the basis of the automated path length correction in more detail in just a moment. While most investigators are familiar with pipetting into the various vessels, including microfuge tubes and cuvettes, the microvolume plate is a bit different. It requires a minimum volume of 2 microliters that is spotted onto a hydrophobic surface, forming a nice rounded droplet. This can be performed using either a single channel or multi-channel pipetter, depending on throughput needs and the assay being performed. Given the minimal volume being used, a multi-channel pipetter is preferred to prevent long exposure times to ambient air, which may result in evaporation and thus concentration of the sample, which can result in increased error between individual samples. Additionally, when performing assays requiring reagents being added to the samples, using a multi-channel pipetter can help to ensure that the reaction times are equivalent across all microspots. I'll discuss specific applications in greater detail in the following sections. Once the apparatus is closed, a nominal 0.5 millimeter path length is created. Within the software, calibrated path lengths are applied to all the sample, blanks, and standard measurements for normalization. The Take3 microplate is compatible with a range of BioTek microplate readers, which offer a variety of different read modes, including our cell imaging line of multi-mode readers. And this allows the flexibility to run a number of biochemical and cell-based assays with a single instrument, with the ability to gather endpoint measurements or generate kinetic data. These include the citation line of imaging instruments, including the citation 1, 5, and the synergy line, including the HTS Neo 2, H1, and HTX multi-mode readers, as well as the Epoch and Epoch 2 absorbance spectrophotometers. As an example, if we look at the Epoch 2 in combination with the Take3, we have a system that can perform many basic applications, such as nucleic acid quantification at microliter volumes, as well as the ability to use cuvettes and microplate-based applications for about the same cost as a single-channel nanodrop, which is limited to microvolume analysis alone. Here's a table of some examples of the broad application space which is accessible when using a multi-mode imager. It's clear here that nearly limitless in the avenues that you can use to approach your research, including some of the more basic applications, cell proliferation, colorimetric assays, ELISAs, as well as some more complicated assays such as gene reporter assays and toxicological studies, as well as the ability to run a number of imaging applications such as cell counting and cell migration assays. If we take a look at the application space for absorbance applications, we can see that even with a less complex system, such as the Epoch absorbance spectrophotometer, when used in conjunction with the Take3 plate, there is a broad range of assay technologies accessible. These include many of those previously mentioned. However, this is a more affordable system with a small footprint. Let's continue our discussion surrounding absorption spectroscopy and turn our focus towards biomolecular quantification. The biomolecules come from a number of sources and can be used in a wide variety of downstream applications. One physical property they all have in common is the ability to absorb light in varying degrees depending on the molecular structure. The physical characteristic can be exploited for quantification as well as to provide additional information regarding sample quality. We'll first focus on the absorption in the ultraviolet or UV range. So the basis of absorption spectroscopy is the ability of the analyte of interest to absorb incident light of a specific wavelength. For example, nucleic acid quantification can be performed using incident light at 260 nanometer, which is absorbed by the nucleoside ring structures, both the purine and pyrimidine rings. The actual measurement is the amount of transmitted light that passes through the sample, referred to as transmittance, as compared to the reference source. This value is calculated and expressed as an absorbance value, which most folks are familiar with. It is important to note, however, that direct quantification can be performed in a non-destructive manner, allowing preservation of the sample. The primary advantage of this method is simplicity, while the primary disadvantage is the limited detection as compared to other more complex analysis methods, which we'll discuss shortly. Also important to note is the requirement of a UV transparent vessel to limit interfering background absorbance. The basis of this determination is the Beer-Lambert law. So while I won't cover the Beer-Lambert law in excruciating detail, it is important to note the difference between absorbance measurements, which conform to this law, showing a linear relationship between absorbance measurements and analyte concentration, as opposed to fluorescent measurements, which may not necessarily show a linear relationship, but can provide lower detection limits. We'll cover this in more detail in later sections. So it's easy to see from this equation that the absorbance is related to both the characteristic molar absorptivity of the analyte, path length through the sample, and concentration of the sample. When solving the equation for the concentration, given a fixed path length and molar absorptivity constant, one can easily determine sample concentration once an absorbance measurement is taken. Let's discuss how we can exploit this relationship for automated path length correction, where the path length through a sample can vary depending on the sample volume, such as is the case in a microplate well. So to correct the path length to 1 centimeter, BioTek's reader control and data analysis software, Gen 5, has a built-in method for samples diluted in water. A stepwise procedure contained in the Gen 5 method is shown here. In step 1, Gen 5's method uses an absorbance peak of water at room temperature, which is 977 nanometer, and a blank measurement at 900 nanometer. The 977 minus 900 optical density differences is then divided by 0.18, which is the known optical density for water at 1 centimeter. The result of this calculation is the path length of the sample, which can then be used to calculate the concentration of nucleic acid in the microwells. In step 2, the sample absorbance measurement at 260 nanometer is divided by the sample path length to obtain a corrected OD value. Then you'll see in step 3, we use the mass extinction coefficient of double-stranded DNA, which is 50 micrograms per mil per OD. And this is multiplied by our corrected OD to determine the concentration of the sample. The analysis method can be extended for use with other biomolecules and solvents as long as the extinction coefficient is known and the solvent has a unique absorption peak. So as I just mentioned, the automated methods can be applied to other nucleic acids and can be combined with several other measurements to provide additional information. For instance, it's common to simultaneously perform absorbance measurements at 260 and 230 nanometers and or perform spectral scanning measurements to gain additional insight into sample purity prior to performing downstream applications. So Gen 5 software allows the inclusion of buffer blanks and sample dilution factors to the plate mapping to automate many of the necessary calculations. Both the microvolume and microplate format are great for collecting multiple measurements from multiple samples without extensive sample handling. Here's an example of the typical performance that can be expected when performing DNA measurements using a cuvette and a biocell with a Take3 plate. You'll notice equivalent performance between the two vessels down to low nanogram per microliter concentrations. Again, these are fixed 1 centimeter vessels. The main difference is the standard cuvette must be capped or sealed and laid on its side to gain that vertical path length versus the vertical path length of the BioSpa with the opening on the top. When comparing microvolume measurements, we also see a nearly perfect correlation with the BioCell, indicating accuracy down to the low nanogram per milliliter concentration. The difference here is the detection limit is approximately 20-fold difference, is due to the approximate 20-fold difference in nominal path length, which is 0.5 millimeters for the Take3 microwells versus the 1 centimeter path length for the BioCell. Shown here is one additional comparison across various vessel types, volumes, and instruments. Measurements are representative of those taken of a DNA sample, which was purified from CHO M1 cells using a QIAGEN All-Prep RNA DNA kit. And we can see all accuracies are within 2% as compared to a standard cuvette, which is considered to be the gold standard measurement. The limit of detection of the sample was determined to be approximately 1.2 nanograms per microliter based on signal to noise of the blank measurements taken. So now let's focus a little more on purity assessment. So the assessment of purity of a nucleic acid sample is often performed by a procedure commonly referred to as the A260-A280 ratio, or just 260-280 ratio, which refers to the two spectroscopic measurements made of these at these defined wavelengths. For double-stranded DNA, the commonly accepted commonly accepted average extinction coefficients at 260 and 280 are 0.2 and 0.11 nanograms per microliter per centimeter, respectively. Similarly, for proteins, the average extinction coefficient values at 260 and 280 are 0.0057 and 0.001 nanograms per microliter per centimeter, respectively. Thus, it's obvious that nucleic acid samples would be expected to have a higher absorbance at 260 than at 280, while for protein samples, the converse would be true. And using these extinction coefficients, pure nucleic acid samples would have a ratio of approximately 2, while proteins would be approximately 0.57. Samples that contain a mixture of protein or DNA would, of course, be influenced by both macromolecules. And according to Beer and Lambert law, the total absorption of a solution is the sum of those absorbencies of the components that comprise the solution. Thus, in the case of a mixture of nucleic acids and proteins, the following equation would be true. Just shown here at the bottom left. And note that since the ratio is taken of absorbencies, the path length variable is canceled out, providing the same vessel is used for both measurements. In some instances, additional information can be obtained by performing a spectral analysis of the sample. Typically, biomolecules are scanned between the wavelengths of 220 and 300 nanometers in 1 nanometer increments. And spectra covering this range will ensure capture of the absorption profiles of biomolecules as well as those of any potential contaminants, should they possess a chromophore of significant absorptivity over this wavelength range covered. Therefore, spectral analysis can provide a relatively quick method of detecting the presence of contaminants without destruction of the sample. So how can we do this using a microvolume sample? So if we look in Figure 1, we can see that, which is on the left, We can see that microvolume spectral scanning analysis representing duplicates of 2 microliter samples of double-stranded DNA in buffer, and this was performed using a Synergy H4 and a Take3 microplate. And the data indicates the ability to perform spectral analysis on these samples down to the low nanogram per microliter range. The values shown here represent 2 microliter samples normalized with blank data, and they've been normalized to a 0.5 millimeter path length. We look on the right-hand side, figure 2, we see biomolecular spectral data comparison. Here are the typical absorbance profiles overlaid of purified double-stranded DNA, which is shown in the blue, BSA protein, shown in red, and then a combination, shown in green, and a combination of the two shown in red. So now if we look at absorbance spectra of purified protein or a mixture at different ratios, this is now looking at herring sperm, double-stranded DNA, BSA protein, or a mixture of the two. And we can see spectral analysis here of a complex mixture containing ranging from 1 to 10 DNA to protein, all the way to 100% protein. And we can see here the differences at 260 nanometers, and this will again lead to the differences that we see in our ratio and allow us to calculate purity. If we look at the right-hand image, again we can look at those different ratios and how they change over the wavelength, and we can see significant differences. Again, these measurements were taken at 1 nanometer intervals ranging from 240 to 290 nanometers. And again, they're all normalized to 1 centimeters. The samples are of purified DNA, protein or DNA mixture, 60-40. And all the ratios were calculated by dividing each wavelength by the A280 nanometers. So if we look at our A260, A280 ratios plotted versus percent DNA, we can see samples here with increasing concentrations of BSA ranging from 40 to 100%, show very little change in our A280 ratio, which indicates that it takes quite a lot of protein contamination before it's reflected in the ratio. The calculated percent accuracy determinations from spectral data shown in the right-hand table reflect this as well. The accuracy of DNA and protein concentrations were calculated using peak absorbance measurements at 260 and 280 nanometers respectively, and the extinction coefficients of 50 nanograms per microliter and 0.667 micrograms per microliter for DNA and protein respectively were used for comparison and calculation of the determinants at the expected values, and these are presented as a percent accuracy. And it's important to note that while it takes relatively little DNA contamination to affect the calculated protein concentrations, considerable protein can be present for affecting the calculated DNA concentrations. And this is important for downstream applications when purity is essential. So a common method to ensure sample integrity when purifying nucleic acids is to treat the source samples with a denaturant to remove ubiquitous nuclease activity. And guanidine hydrochloride at a stock concentration of 8 molar is typically added to lysis or homogenization buffers during the sample preparation. Residual guanidine hydrochloride at trace levels, which can be less than 1%, can impact quantification of DNA due to an apparent decrease in the spectral absorbance peak at approximately 260 nanometers. And this may be due to the chaotropic effect of the reagent, causing partial denaturation of the DNA, and appears to be independent of concentration. Phenol provides a second example of a commonly used reagent in molecular biology for the isolation of DNA, RNA, and proteins. The method is commonly referred to as phenol chloroform extraction and relies on a 1:1 ratio of water-saturated phenol to chloroform. And precipitation of nucleic acids is generally performed subsequent to extraction for removal of residual phenol. And here we show the calculated DNA concentrations from spectral data. Phenol concentration. DNA concentrations are calculated using the A260 measurement and an extinction coefficient of 50 nanograms per microliter per OD in the presence of the specified concentration of contaminant. It's obvious from this plot and the table that even trace amounts as low as 0.2% volume of residual phenol can significantly impact accurate quantification of DNA. Now I'd like to change gears and discuss applications using fluorescence spectroscopy. So while UV absorbance measurements methods used for quantification of nucleic acids in solutions can provide information about the overall abundance and purity, it's impossible to discern the relative abundance of DNA and RNA present in a sample when they are together. And since detection and accurately quantifying DNA is important for a range of downstream applications, we need a more precise and discriminatory method for that quantification. So picogreen is an extremely sensitive asymmetrical cyanine dye that can be used for DNA quantification. The unbound dye exhibits no intrinsic fluorescence, but upon binding to double-stranded DNA, the dye fluoresces strongly with a maximal emission of approximately 530 nanometers, which can be seen here, and provides up to 1,000-fold more sensitivity than UV absorbance methods. It's highly selective for the double-stranded DNA over other varieties such as single-stranded DNA and or RNA. So here's a standard microplate-based assay protocol where herring sperm can be diluted. In this case, we dilute it to 2 micrograms per mil in TE buffer. The final concentration is confirmed using 260 nanometer absorbance, and then a series of dilutions ranging from 0 to 2,000 nanograms per mil can be generated. And then we would aliquot 100 microliters into each microplate well, and that should equal, or we add equal amounts of picogreen quantitation reagent. The mixture is then incubated for 10 minutes at room temperature, and we want to protect that from light. And then we can read the plate on a microplate reader using the appropriate wavelengths, which are excitation of 485 nanometers and emission at 528. So if we look in the upper left-hand corner at the plot of the fluorescent excitation and emission spectrum of double-stranded DNA, this spectrum was taken on a sample of 1 microgram per mil double-stranded DNA and TE stained with picogreen reagent. And this data has been normalized such that the maximum value was 10,000 RFUs. And we can see our peaks around 450 nanometer and 525 nanometer for excitation and emission respectively. And then if we now move to the upper right-hand corner, look at the plot of linearity of spectral filter-based detection, we can see we have excellent linearity in the concentration range from 0 to 1 microgram per mil. And The insert figure depicts data points for the lower concentrations, which range from 0 to 1,000 picogram per mil. And we can see we have excellent correlation here using least mean squared linear regression analysis, showing near perfect linearity. If we then go directly below that, we see linearity of monochromator-based detection, again showing excellent linearity. And these concentrations are the same concentrations as shown above. Again, we can see near perfect correlation. Then if we go to the lower left, we can look at a comparison of the signal to background ratios of those measurements determined using monochromator versus filter-based optical pathways. And I think it's obvious here that we get a slightly better signal-to-noise ratio when we use the filter cubes. So now I'd like to discuss picogreen in situ microvolume assay, and I'd like to discuss the protocol, and then I'll discuss an application based on that protocol. So with the microvolume protocol, this can be performed on the Take3 microvolume plate and requires the addition of 2 or 3 microliters of your sample added with an equal amount of reagent, in this case 2 microliters of sample and 2 microliters of reagent. And then once those are added and mixed, we close the Take3 lid and incubate at room temperature for about 5 minutes. And this is a much more economical assay versus the microplate assay, where our reagent and sample volumes are significantly reduced, approximately 50-fold. And you can see we have excellent linearity regardless of whether we use spectrofilters or monochromators. This data is shown in the lower right-hand corner. It shows we can get down into the low nanogram per mil range and have excellent linearity. So automated quantification of nucleic acids in microplates has been performed for decades, yet the challenge presented here for this application that I'd like to show is to develop a higher throughput miniaturized assay of double-stranded DNA using a low-volume microplate, such as the Take3. However, we needed a higher throughput, so we needed a much higher density, and in this case we used a UV Star Microclear 384 well microplate from Griner Bio1. And this plate is clear bottomed, and however it has it's black walled and is free of detectable DNAs, RNAs, and human DNA contaminants. The high density and the shallow well feature, 20 microliter max volume, offers the ability to significantly reduce analysis volumes while maintaining sufficient path length for accurate and precise measurements of double-stranded DNA. And part of this analysis was to incorporate the use of both UV absorbance analysis with the sensitivity of the picogreen assay. So first, to determine the minimal volume required for precise optical density measurements in the UV Star plate, we performed a 1:3 serial dilution of 10 mg per mil of double-stranded DNA, and that was pipetted at either 2 or 4 microliters in replicate wells. And Table 3 here demonstrates Gen 5 calculated double-stranded DNA concentrations from optical density measurements in addition to the precision data. The path length using 2 microliter total volume was determined to be 0.6 millimeters for 4 microliters, for 2 microliters. For 4 microliters, it was 1.2 millimeters. And it's evident from the precision data in Table 3 that significant improvements in precision can be obtained by the use of 4 microliters. And this is entirely due to the increased path length. And this improved precision is comparable to larger sample volume determinants measured in a 96 well microplate. And it was comparable up to approximately 1.5 micrograms per mil. So following determination of the minimum sample volume required for analysis, a series of experiments were performed to validate the method. A dilution series of double-stranded DNA was created and quantified via cuvette and the UV star plate for direct comparison. Quality assessment of the DNA by cuvette was performed using a Shimatsu PharmaSpec, and it revealed that the A260-280s and A260-230 ratios were consistent over much of the dilution series and met the commonly accepted values for purity of nucleic acid as seen in the table. At concentrations below approximately 50 micrograms per mil, the ratios declined significantly below the commonly associated values, which are indicative of a pure sample, that should be approximately 1.8. And this is a common observation for diluted nucleic acid samples. So here is a workflow diagram for this particular assay. Sample IDs can be imported into Gen 5 data analysis software. We will then perform measurements on a blank empty plate for background subtraction. We'll then pipette 4 microliters of our standard ore sample into the plate and read the plate at the appropriate wavelengths, 230, 260, and 280 nanometer, as well as collect data at 320 nanometers, which will detect any plate abnormalities. We'll then add 20 microliters of picogreen reagent to all the wells, and after the appropriate incubation period, we'll read the plate with using 488 nanometer excitation and 530 nanometer emission. And then Gen 5 will automatically estimate the DNA quality and concentration. Accurate estimation of the concentration based on the picogreen dye is shown up on the in the upper left-hand corner, where we've plotted concentration versus RFUs. And if we look at the table, we can see a comparison of those concentrations to those taken in a microwell or a cuvette. You can see that we have a dynamic range of approximately 60,000 RFU in the typical working range of sample concentrations. And furthermore, we can see our CVs are quite low, ranging from 0.9% to 6%, which are indicative of good correlation between replicates. And again, if we look at our plot versus RFU, we see excellent linearity up to the high hundreds of nanograms per microliter, which is again in the typical linear working range. This assay can be modified based on the concentration range needed by the customer. In this case, we were using the lower range up to several hundred nanograms per microliter. Now I'd like to discuss a few tips and tricks to help minimize contamination and increase accuracy and precision regardless of method. It's very important to use gloves to protect samples from nuclease and or other contamination which may be found in the human skin and can skew our results. It's also very important to ensure that the sample vessel, whether it be a cuvette, microplate, or the microvolume surface of a Take3 plate, is absolutely clean, especially when working near the lower level of detection limits, which can be skewed by even 1 or 2 milliOD. So we also want to ensure that our instrument is well maintained. Again, contaminant on any of the optical components can skew the results, or contamination of the detectors themselves. We want to ensure that we use replicate samples whenever possible to reinforce our accuracy. Duplicate or triplicate samples are recommended, again, especially when working towards the lower limits of detection. And when we're working with low volumes, it's going to be extremely important to limit exposure to the air. with a sample of 2 or 3 microliters, exposure for only a few minutes can have significant changes, up to 5 or 10% of accuracy. Also, again, when working at low volumes and multiple samples, we want to try to use a multi-channel or repeater pipetter, especially when spotting samples and or adding reagent to those samples. And again, blanking is very important. We can do this in several different manners. We can either use our buffer to blank, or we can use an empty vessel, or a combination of both. Again, this is going to be extremely important when working near the lower limit of detection, as well as when using very, very small volumes. So in conclusion, just like to reiterate that multi-volume analysis of biomolecules is possible due to the flexible nature of microplate instrumentation, where a variety of different vessels are available to perform those different assay types. And there's a wide variety of compatible assay methods that can be coupled with the various detection modes that are available on these multi-mode instruments. And we can do a wide range of sample concentrations as well by using different volumes. Spectral analysis, especially when applied to biomolecular samples, provides additional information that can help determine sample purity prior to performing any downstream applications, while the microvolume analysis itself provides a method to preserve precious sample and conserve reagents. I'd like to thank you all for attending again, and we'll take any questions at this point. Thank you.
Thank you, Peter, for that terrific presentation. As you mentioned, we'll now take a look at some of the questions that came in. If you've not done so, please be sure to enter your questions in the chat window in the bottom left-hand corner of your screen.
Okay, so we have a few questions here. So one question is, can you tell us about differences in quantification that are seen when comparing A260 and picogreen measurements? So I tried to address that in the application, and essentially the basis of the application towards the end of the slides were to use the absorbance measurements, the A260, A230, and A280 measurements to determine purity based on those common ratios that we see, the 1.8 or 2.0 ratios, and that can be applied to both DNA or protein samples to assess the purity, whereas the picogreen measurements allow us to lower that detection level. And essentially what I showed there was a range of concentrations, and then we can use those picogreen RFU plots versus concentration as a standard curve to back calculate our sample concentrations, and this allows us to extend that range. Second question, do you have 96 well micro spot plates? We have a 48 spot plate, essentially the Take3 Trio, which I believe was shown in one of the earlier slides. The Take3 Trio has 3 slides in it, and each one is 16 spots, so that gives us a total of 48. However, the trade-off is that it does not have a spot for the cuvette and the biocells. Those are taken up by the additional slides. However, you could have both in your lab, and that would allow you to have both of those options. An additional question, what is the minimum total volume for a 96 well plate format? So that depends. There's several different types of 96 well plates. There are some low volume plates that allow you to go down to tens of microliters. A standard 96 well plate, if used for DNA quantification, would be approximately 50 microliters, and that allows you to have a path length that is long enough for that determinant. If you start to get lower, then you run the risk of having a very, very short path length. Another question, how could I test a sample that consists of single-stranded DNA versus double-stranded DNA? Looks like this person had used some other dyes that bound to both. So I believe Life Technology has some specific dyes that can be used. I don't have that information at hand, but that's certainly I would contact those folks. We've worked with several of their dyes, and I think that they provide a nice array of dyes. So that would be my recommendation. Another question here, what is the A260, A230 ratio typically used for? Again, this is a ratio that can be used to look for contaminants where, and I believe I show some information here on a couple of these slides when we're looking at the purity of a sample. And so we can use either the 260 or 230 ratio depending on where the absorption peak is of the contaminant. And so a lot of folks will use that as a secondary measurement in conjunction with the A280 measurement as well. Let's see. There's another question here, can we measure double-stranded RNA using the Take3 function? We haven't done a lot of work here with RNA just because we're not set up as an RNA lab, and nuclease contaminant can severely limit the ability to perform those types of assays. However, there's no reason why you can't use RNA or you can't use a Take3 for RNA quantification. You'll just need to adhere to the standard protocols for cleanliness when working with those RNA samples to ensure that you're accurately quantifying those. We have another question here, quantification of oligonucleotides with an Epoch, asking for a protocol. This is something that I can certainly help with offline, so why don't we hold that and I'll respond to that question a little later. Another question, when using picogreen, do we need to take measurements at two wavelengths, 480 and 530? So you're using the 480, that would be 480 nanometers your excitation wavelength, and then you're actually taking the measurement or the reading at 530, which is the emission from that picogreen fluorophore. Another question, is the Synergy 2 capable of DNA quantification? Absolutely. So the Synergy 2 should have the capability to read absorption using the monochrometers as well as fluorescence, depending on the model of the unit. If it has all the modules, we can certainly help you with that as well. If you're unsure, you can call into our Technical Assistance Center. You can look at your model number and help you with that. So there's a question regarding the Synergy Neo plate reader as far as hardware, particular for measuring 4 microliters. So not necessarily, although some of our instruments have slightly different optics. Certainly the Synergy Neo is capable of measuring 4 microliters in a 384 well plate. Again, recall that these are special 384 well plates that are a little smaller in diameter, essentially the area. They're half area plates, which give that higher or longer path length at the smaller volumes. So there is an application note on our website which discusses the higher throughput method that has the part number for that plate from Griner, and I'm sure there are others out there as well. It's just important to make sure that that's a UV transparent plate if we're performing the absorbance in the UV range. For Pico Green, you have more flexibility in the plate type that you use because it doesn't necessarily need to be UV transparent. I think that's all the questions we have time for right now. I really appreciate everyone's time today, and feel free to send in any additional questions if you have any. Thank you.
Thank you all for joining us, and thank you to our presenter, Pete Brescia. For more information on products featured, we encourage you to visit our website at www.biotech.com. If there are any questions that we weren't able to get to, we will be sure to get back to people via email. And as a reminder, you'll receive a link for this webinar as soon as it becomes available. We hope you will join us again and have a great rest of your day.